Welcome to chromatography. Chromatography is a century-old technique which can be used both to separate a mixture and to identify its components. There are many variations of chromatography, but they all involve the same two basic elements. There is a stationary phase which does not move, and this is either a solid or a viscous liquid. And there is a mobile phase, which is a liquid or a gas, along which the components of the mixture move. We have here a schematic representation of a chromatography experiment. The white area represents the mobile phase, and a fluid is moving in the direction of the arrow. The blue area is the stationary phase. That is, the stationary phase is the one which does not move. We are putting on a mixture of the green and white molecules at the beginning here, and now we will start the flow of the mobile phase. The white molecules are more strongly absorbed by the stationary phase, whereas the green molecules seem to favor the mobile phase. In the end, the green molecules, which kept their nose to the grindstone, ended in front, and the white molecules, which stopped to smell the roses, lagged behind. But the important thing was that they were separated. In the experiment you'll be doing, the stationary phase is going to be paper. It's going to be something very similar to filter paper. The mobile phase will be a mixture of water, hydrochloric acid, butanol, ethanol, and acetone. Now that may seem very complicated, but each one of those components has its purpose. The stationary phase is the paper. Now to prepare the paper, what you do is you draw a line about 1.5 centimeters to 2 centimeters from the bottom. This is the spotting line. We'll divide that up into individual little spaces where you will put the individual knowns, silver, cobalt, etc. You will have a mixture of all 5 knowns here, and then you'll have 2 spots for your unknowns. The rationale of this is that both your knowns are duplicated by the individual ion and as in the mixture, and then also your unknowns are duplicated. So you have 2 results for every ion which you're dealing with here, and the 2 results should agree with each other. Well, we're all set to start our lessons by chromatography. We have a chromatography paper here all set up for ourselves. We have a capillary tube that we'll use for spotting. We have the knowns here, including one containing all the knowns, and the unknown. We have a beaker which will carry out elution. We have water here so that we can wash out the capillary tube between spotting. In spotting your chromatogram, you will use a capillary tube. Now you see, if you put the capillary tube into a liquid, it draws up not only to the level of the liquid, but more than that. And then when you take it out, then you'll have enough liquid in there to spot your chromatogram. In spotting your chromatography paper, you have a capillary and you put some of the solution. This is a 10th molar copper solution. And then you just touch it to the point where you want the spot to form. Hold it there until the spot is about 1/2 centimeter or so wide, and then just remove it. Rinse out your capillary between solutions and shake it dry. Now remember, you've put about the same amount of material on all of these spots at this point, which means that since these ions over here were pure ions, they contain more of that individual ion than the spots over here, which probably are mixtures of ions. For example, the known has about 1/5 as much mercury in it as the mercury which had just pure mercury in it. So what you will need to do is you need to take and you need to put a second layer on this known. That's called double spotting, and you do that after the first spot has dried. Otherwise, instead of just making a higher concentration, you'll just make a bigger spot. You are now set for elution. What you must first do is make a cylinder out of your chromatogram. You do that by scotch taping the two ends at the top here. Notice that they're not exactly touching so that solutions from one side do not drift over to the other side. This will then fit into your 600 milliliter beaker. You're all set then. Put in your eluting solution. Put in your chromatogram carefully. Cover it over with a piece of plastic wrap and secure it down with a rubber band. And then you can let this sit until the chromatogram has been eluted. Be careful, don't make waves. When the eluting solution gets about 2/3 of the way up the chromatogram here, you can take the chromatogram then out of the eluting tank and start developing it. The first thing that you must do is mark the solvent front line here. How far has the solvent risen on the chromatogram itself? You can do this in ink. After you have marked the solvent front line, or the distance that the solvent has traveled on this chromatogram, then you can look for spots which you can see. I've seen some yellow spots here which I have circled. Now note, I can use a pen in doing this, but I'm using a towel so I'm not contaminating the chromatogram. Next, we'll put the chromatogram in the ammonia tank and let it sit there. There'll be changes taking place here. More spots will show up. When all the spots have shown up, then again we'll take it out of the tank and mark those spots with circles so we know which ones showed up and what the colors were when we put it in the ammonia tank. Here's what we've seen so far, and we've circled all the spots which are showing after the ammonia tank. We'll now put it in the hydrogen sulfide tank, and as you can see, we have a lot of dark spots showing up. And so here is our fully developed chromatogram. It has all the ions in it, and as you can see, the unknown seems to have two species. One appears to be cobalt, and the other appears to be the mercuric ion. In paper chromatography, the distance an ion travels is given in terms of something called a retardation factor. That is the distance the ion moved relative to the distance solvent moved. And thus, in different chromatographies, you will get the same values. So, the way this works, this is a spotting line here. This is the starting point where the center of the spot was, and the spot moved up this far. The center of the spot is here, and you measure this distance. The solvent, meanwhile, started at the solvent line and ended up at the solvent front line, which we drew in. The retardation factor for the cobalt-2 ion is given by the ratio of the distance the ion traveled relative to the distance that the solvent traveled, and it has the value of 0.434. Now note, distances have units, but the retardation factor is unitless because it is a ratio. And now a word from our sponsor, chemistry. To take the mystery out of chemistry, read your manual and do your pre-lab. You know, cobalt and copper are a couple of very colorful characters, so you'll see more of them later this year. So what do these greenish-black iron sulfide spots have to do with hard-boiled eggs? Here are two hard-boiled eggs. The one on the right had been boiled for about 20 minutes. The other was boiled for about half that time. Now the whites of the egg consist of sulfur-containing proteins, and these decompose into hydrogen sulfide upon heating over time. Now the yolk is rich in iron, and the hydrogen sulfide diffuses towards the yolk, and over time it forms the same iron sulfide that you found in your chromatograph. Here's another question. In the eluting, why does the silver ion barely move? The silver ion is really very soluble in water, but remember the eluting solution has hydrochloric acid, and this is what happens when hydrochloric acid meets with the silver ion. Remember that rusty brown spot you got when you put iron into ammonia? Well, that's exactly what it was. Rust. Ferric hydroxide. And now we'll return back to our final feature, the real truth about black ink.